You already mentioned my title. I will talk about human biomonitoring, chemical exposures. First, some introduction on exposure assessment in a classical way. Classical exposure assessment in the past has been performed by ambient monitoring, measuring chemicals in the environment, air, water, soil, consumer products, and then combine all these sources, do model calculations, cumulative exposures, and then associate these external exposures to health effects for exposure and risk assessment. Since, I would say, 30 years, human biomonitoring found its place next to ambient monitoring. Human biomonitoring measures internal exposures in urine, blood, or other body fluids mainly. And there are three different varieties of human biomonitoring, depending on what exactly you measure. If you measure the parent chemicals and the metabolites from these chemicals, you call it exposure biomonitoring. And if you go further, like reaction products of chemicals with body macromolecules or disruption of hormone levels, then you would call it biological or biochemical effect monitoring. I will talk here only about exposure biomonitoring and which you can use to compare it with external exposure assessment to check and validate such models, or going the other way to look at biomarker levels and health effects in epidemiological studies, or to compare the biomarker levels with limit values. One of the big populations or population studies we have in Germany is the German Environmental Survey. It's a population representative sampling of around 2,500 individuals. You see the map of Germany here to the left. In the center, in the heart of Europe, and you can see the 167 cities and communities that are used to sample these individuals. Sampling is performed by home visits, interviews are performed. You also collect tap water, house dust, and air, but during these visits you also collect blood and morning urine. And with this German Environmental Survey, you get population representative exposure distributions, and of course with the questionnaire and the interview data you can also perform exposure source investigations. The most recent German Environmental Survey, or GERES, has been GERES 5, performed in 2014 to 2017 on children and adolescents, and the GERES 6 has been planned to start in 2019 but has been postponed due to obvious reasons. That one was intended to be centered around adults. Another important source of biospecimens is the Environmental Specimen Bank. It's a continuous collection of human specimens since the early 80s in Germany. We have 4 sampling locations. You see them on the map here, 2 in former Eastern Germany, 2 in former Western Germany, and 120 university students are sampled each year at each location, and the distribution is 50% males and females. And what's very important to know, we collect 24-hour urine samples and accompanying blood and plasma samples. You can see how these samples are stored. They are stored in a bunker in Munster. It's a these samples are considered a national heritage and the treasure, so they are kept very, very safe. But we have the chance to get some aliquots to perform biomonitoring measurements. And the beauty with these samples is you can also perform a retrospective time trend analysis, because you have these very old samples and you can apply new methods on them to measure exposures in the past. And because it's they are 24-hour urine samples, you can perform very robust daily intake calculations and a robust risk assessment. Uniting all these two populations, since 10 years, we have the so-called BMU VCI human biomonitoring cooperation. It's a cooperation between the Environment Ministry and the German chemical industry. Since the aim of this project is to select 50 substances of high interest, 5 each year starting from 2010. These substances are selected by university experts, industry experts, and agencies. It can be new chemicals that are newly introduced, or it can be legacy chemicals which have not been able to be measured by human biomonitoring. So for these 50 substances, we intend to develop new analytical methods and identify new and valuable exposure biomarkers, and then apply these new methods in the population studies I just pointed out to you. The very interesting feature of this cooperation is the financing, because it's a joint financing between the German Environment Agency and the German chemical industry. So the chemical industry finances the analytical method development, while the Environment Agency finances the measurements in the population studies. All of this is also accompanied by the German Human Biomonitoring Commission, which derives limit values for these new chemicals. And also, if the population study data is there, derives reference values. These are the substances that have been selected. Don't worry, I'm not going to talk about all of them, but you can see them here. The ones underlaid in blue are developed in my laboratory. And in light blue, I have collaborations with partners developing these methods. Just as a quick run through, the boxes in red point out plasticizer substances. Boxes in green, disinfectants. In black, these are fragrance materials. In orange, you can see UV filters. And in violet, you can see solvents and detergents. In one example, I am talking about the phthalate plasticizers and substitutes. You all know, and Leonardo Trasande already pointed it out, they are high production volume chemicals. They use as plasticizers, solvents, additives. And the big problem is several of the phthalates are known endocrine disruptors, and the general population is known to be highly exposed to them. So what is the consumption in Europe? You can see that the main plasticizer DHP has dropped rapidly in consumption because of the endocrine disrupting effects. The product use has been limited, and also other endocrine disrupting phthalates have been dropping, while some other phthalates have been increasing. So you can see a substitution within the phthalates, but also totally new substitutes have come on the market. The question now is, how do they reflect in the population exposures? This is the consumption data you see here. So for the phthalates, for the classical phthalates, we already have established biomonitoring methods available. And for the newly introduced plasticizers, we developed new methods in this cooperation project. This is now a slide on biomonitoring data in the environmental specimen bank, and you can see I show you the data on DHP measuring the hydroxylated metabolite, and you can see the years we have been sampling in the environmental specimen bank. So look at the logarithmic scale, and you can see the drop of the internal exposure levels of the urinary concentrations from the early 90s to last year. It's a drop by more than a factor of 10. It's a logarithmic scale, so it's a factor of 10, and it's very similar to the consumption numbers. So that's the specimen bank data here. As I pointed out, we can also relate this to the toxicological limit value. It's the HBM guidance value. It's the analog of the tolerable daily intake, and you can see that in the last millennium, we had exceedances of this limit value in the population. Here you can see the blue box. It's the data from the German Environmental Survey. So this blue box here is on 2,500 children. And you can see one feature is that, remember, the white boxes are the German students, 29 years old, that the Cherres children are approximately 5 times higher exposed than the young students from the environmental specimen bank. That's a general feature of phthalates. Phthalates show higher exposure in children than in adults. This is specimen bank data for another phthalate, dibutyl phthalate, measured via the monoester metabolite in urine. You can again see a dramatic drop in exposure, this time 25-fold exposure drop. If you look at the HBM guidance value, you can see that in the last millennium, more than 50% of the population of the young Germans exceeded the tolerable daily intake. But now in the last couple of years, we have no exceedances for this phthalate anymore. So now, and again, you can see that for the environmental survey data for the children, that the children still have higher exposures than the young adults, again, approximately a factor of 5. And in the children, you can still see exceedances of this tolerable intake level. Now let's look at the substitute sites. Remember those that have been newly introduced, and we developed methods for the terephthalate. You can see here measuring a carboxylated metabolite in urine. You can see the rapid increase in exposure to terephthalate starting in 2010-11. In 2016, each and every sample analyzed was positive for terephthalate metabolites. All that has been published recently. This is the specimen bank data. And we see, if we look at the HBM value, that's again the tolerable daily intake equivalent, we are still quite far away from it. So this exposure seems to be acceptable right now. However, you can also see, again, same feature with the phthalates, the substitute is the same feature. Children seem to have higher exposures than the young adults. And now if you look at the US, this is data from NHANES, you can see that the Americans are have 10 times higher exposures than the Germans. And you can see the same characteristics again. Children have higher exposures than adults. And if you look at the upper whiskers there, you can see that they're already pretty close to the tolerable intake levels. Another substitute, DINCH. DINCH was introduced in 2002 by BASF, and you can see that we first measured it in 2006. In 2013, every sample analyzed has been positive for DINCH metabolite hydroxyminch, but the increase has somehow leveled off. Up here, you can see the TDI equivalent. You can see that we are quite far away from it here in the specimen bank. But again, you can see that the children's of the environmental survey seem to have 5 to 10 times higher exposures to DINCH than the young adults. Again, the data from NHANES. This time you can see the DINCH exposure in the States is not that high compared to the terephthalate, which could be explained maybe that DINCH is a chemical introduced by the German company BASF, and probably the terephthalates are more produced by American companies. It could be an explanation. As a second example, I want to give you some information on nodal phenol. Nodal phenol, you know it probably as a legacy chemical, because nodal phenol is a high production volume chemical that has been used since the 1930s. It is used as a detergent in textile production, cleaning, pesticides, cosmetics, and it's known to be a ubiquitous ubiquitous chemical in the environment, nodal phenol. Problem is, nodal phenol is also known for its endocrine disrupting effects. So the question is, how high are we exposed to nodal phenol? The problem is, because of its ubiquitous exposure in the environment, nodal phenol is everywhere. We have problems analyzing nodal phenol directly because we have high blank values and contamination in the lab. So we investigated the human metabolism of nodal phenol, and we see oxidative modifications at a lot of places in the alkyl side chain and also in the aromatic ring. But we found that the hydroxy metabolite, 1-hydroxy introduced in the alkyl side chain, is a major metabolite excreted in urine, representing 62% of the oral dose. So this is a very valuable exposure biomarker. This one is, contrary to nodal phenol itself, robust. There is no external contamination, and because of its high conversion factor, it has a very high sensitivity. So we used this exposure biomarker to investigate population exposures. Again, this is data from the German Environmental Specimen Bank. We went back to samples from 1991. You can see already there, each and every urine sample we analyzed has been positive for this biomarker. And we see rather stable levels in the old millennium, but the decline starting at the turn of the millennium and going down quite rapidly in the last couple of years. 75% reduction that goes very well along with the regulatory measures that have been enacted to limit exposure to nodal phenol. This is the same slide, but now I translated the hydroxy metabolite into daily intake. So you can see the daily intake here. We can now blend in the provisional TDI of the Danish EPA, and you can see, at least in Germany and for this population, we are quite far away from critical levels. But we have not yet analyzed children samples from the German Environmental Survey. But we were lucky to have samples in our freezers from colleagues from Japan. You can see here, this is data from Japanese children, almost 200 Japanese children sampled from the year 2012 to 2017. You can see very similar effect of decline that we have observed in the specimen bank, and the levels seem to be very similar in this case to the German adults. So maybe children are not additionally exposed to nodal phenol. But we had another set of samples, and they were from Thailand, Indonesia, and Saudi Arabia. This is from a collaboration with dear Professor Choi from SNU. And you can see that here we see quite dramatic differences between the 3 countries investigated in the children there. And you can see that in Indonesia and Saudi Arabia, the exposure to nodal phenol is about more than 10 times higher than in Thailand. Thailand is known to have rather strict restrictions on nodal phenol in the last years. And you can also see that in the upper percentiles here, some of the children from Indonesia and Saudi Arabia reach potentially critical levels here. Of course, we were interested in the sources, and accompanying all this data have been questionnaires, but we found that the potential sources varied by region. They could not be readily identified. And of course, in all these regions, we have an uncertain environmental background. So for now, we can only state that there are considerable regional differences in NP exposure, that is quite a complexity of exposure pathways, and the main sources of exposure next to environmental background exposure need yet to be identified. So let me sum up my slides. I hope I could show you that human biomonitoring is a multidisciplinary approach that requires analytical, toxicological, medical, and statistical experts. But if you do a good human biomonitoring, you can objectify exposure. So contrary to ambient monitoring, you don't have to perform complex modelings, and it's not a worst case approach. It's an integral measure of exposures. It measures all exposure routes and sources. You can compare populations, and you can visualize time trends, and you can also use new methods with old samples. You can perform toxicological evaluations. You can compare it with limit values on the one hand, or you can use biomarker data for association studies with epi studies. And you can objectify the exposure to be later used for regulatory measures. And with that, you can be support for policy in the politics. The big question in the end is, do we see or will we see a pandemic effect? Well, we will see this if we analyze the 2022-23 samples. I am rather sure that we will see effect because the exposure depends on lifestyle, income, and both Leo Trasande and Professor Christakis have shown that there will be a pandemic effect, and I'm sure it will be seen in human biomonitoring. With that, I want to acknowledge my funding sources and my project partners, and a big hello from my HBM group at the IPA. Thank you.